So exercise 19 deals with the blood. So blood, you may not think of it as a tissue, but it is classified as a tissue. It's the only fluid tissue in the human body. It is a type of connective tissue. Components of the blood, you have the living cells, which are known as the formed elements, and then you have that non-living matrix, or the plasma. The plasma, when you look at it, composes about 55% of the blood. The major component of it is water, but it does also contain things like salts, various electrolytes, sodium, potassium, things like that. It does contain some proteins, and there may be some other substances such as all of your nutrients, waste products, oxygen, carbon dioxide, and hormones that are being transported. And then about 45% of the blood is what we call formed elements. These are your cells. And there's different types of cells. You have your erythrocytes, which are your red blood cells. You have your leukocytes, which are your white blood cells. And then you have your platelets. Now your erythrocytes are going to be involved with transporting gases, such as oxygen, carbon dioxide. The leukocytes, those white blood cells, are involved with defense and immunity. And then the platelets are going to be involved with blood clotting. So everybody has their own purpose and function with it. Now when you look at a sample of blood under the microscope, most of what you should see will be your red blood cells, or those erythrocytes. Notice there's no nucleus in a mature red blood cell. So this is what they will look like. These little tiny small purple dots, those are your platelets. And then the larger cells that do contain a nucleus are your leukocytes, or white blood cells. We can distinguish between them by the shape and number of the nucleus that's present in it. There's 5 different types, as we'll see in a moment. So the formed elements, the type of leukocytes, we have a way of dividing them. You know how we like to subdivide everything. So there's 2 main categories, the granulocytes and the agranulocytes. The granulocytes, when you stain them, it looks like there's grains, almost like someone's dumped a bunch of sand in the cytoplasm. They do tend to possess nuclei that are lobed, and this will include your neutrophils, your eosinophils, and the basophils. The agranulocytes don't have those granules that you can see when you stain them. The nuclei tend to look either kind of kidney-shaped, oval, or spherical, and this will include your lymphocytes and your monocytes. So when we look at the formed elements, the erythrocytes, as I said, are going to be your red blood cells. They're transporting the gases, oxygen, carbon dioxide. They are biconcave. They're anucleated. We know from, say, a lab tech standpoint, we know roughly the concentration, how many cells per cubic millimeter would be 4 to 6 million. So they're the most numerous of all of them. With the leukocytes, you have those 5 types in total, the granulocytes and the agranulocytes. So the 3 granulocytes, once again, are going to be your neutrophils, eosinophils, and basophils. So right here you can kind of see that the cytoplasm looks grainy. That's how they get their name. Once again, the relative numbers of them. Majority will be neutrophils. And here the cytoplasm tends to, with the stain we use, tends to stain kind of pinkish color. Very dark nucleus, contains 3 to 7 lobes. Their function, the neutrophils, is that they are very active phagocytes. When you have an infection, the numbers should increase. Eosinophils, their granules are going to look red, so much darker than what you saw with the neutrophils. Their nucleus tends to look kind of like a figure 8 or bilobed, and the nucleus tends to stain kind of a blue-red color. Purpose of the eosinophils, they will kill parasitic worms. They are also involved with allergies. So this is one thing that you could use if you do what we call a differential blood count, where you not only look at the ratio of red blood cells to white blood cells, but you determine what type of leukocytes or white blood cells are present here. How many neutrophils, how many eosinophils. If someone is having an issue with allergies, you would expect the eosinophil numbers to increase. But instead, if they're suffering from an infection, you would expect, let's say it's a bacterial infection, you would expect the neutrophil numbers to increase. So these can all be different clues for you. Like I said, if you had a parasitic worm infection, you would expect eosinophil numbers to be higher. Basophils, they're going to have kind of a darker bluish purple granules, and the nucleus is going to look kind of U or S-shaped and will stain dark blue. So the granules are blue, the nucleus is blue. These basophil numbers will increase or play a role with inflammation. They do contain heparin, which is an anticoagulant. They also release histamine. So this is something else that's going to play a role if you have an infection. Sometimes these numbers will play a role. They will release histamine. What do? You take an antihistamine to counter the effects of it, because when it releases histamine during inflammation, you start to see side effects from that as well. The lymphocytes are one of the agranulocytes. When you stain at the cytoplasm, remember it's not going to have those granules in them. Cytoplasm is going to look like a pale blue color, and you're going to have a very large, dark bluish purple nucleus. It's essentially going to take up most of the cell. Lymphocytes play a role with the immune system. You have B lymphocytes and T lymphocytes. So the T lymphocytes are going to be involved with fighting infections, kind of a direct cell attack, and then the B lymphocytes produce antibodies. So they're both involved with your immune system. The monocytes, which will have kind of a light gray-blue cytoplasm and a dark nucleus that's often kind of kidney-shaped. And the monocytes are an active type of phagocytes. When they are in the bloodstream, they are the monocytes. When they move from the bloodstream out into the interstitial fluid, that fluid between the cells and the tissues, they mature into the macrophages. This is your cleanup crew. Whenever you have an infection, whenever you have any type of injury, tissue damage, those macrophages are going to come in and clean up the debris. They carry out phagocytosis. So it's one of those things. Macrophages start out as a monocyte in the blood, and then when it moves into the interstitial fluid, it matures, if you will, into the macrophage. And then your platelets, they look like little cell fragments. They will stain deep purple, very small as compared to the leukocytes and erythrocytes. And they are involved, as I said earlier, with blood clotting. So those are the formed elements that are found, the cellular components of the blood. When we look at the human blood groups, your blood type is determined genetically. There are certain proteins or markers that will be present. Antigens, when we use from an immune standpoint, we use the term antigen for any foreign substance or anything that the body will recognize as foreign, and then it starts this immune attack against it. It's all for protection. Antibodies are what you make. Now, when we talk about blood typing, it's a little bit different, because when we type the blood, we're going to be using antibodies and we're going to be seeing what antigens, etc., may be present. With the major blood typing, and there's several different proteins you can look for on blood typing. It's what we call major and minor. For most of the time, what you're looking at is just the major. Now, if somebody were to have a tissue or organ transplant, then you've got to do the minor blood typing and look at all the different proteins, and you want it to be as closely matched as possible. But for most things, you're just going to be looking at the major blood typing. There's 4 major blood types with this ABO blood grouping. There's type AB, type A, type B, and type O. So I'm going to use this chart to try to explain this. Let's start with type AB blood. An individual who is type AB blood, they are going to have these. Now here where they use the term antigens, sometimes I kind of hesitate to use that because you think of something foreign. Think of it as you have these A and B markers. Where are they located? They're on the surface of the red blood cell. So that's why it's shown here as this triangle and circle shape. So the markers that's on the red blood cell are both A and B. What type of plasma antibodies do you have? You don't have any. What type of blood can this individual receive? They can receive type A, type B, type AB, and type O. So they are known as the universal recipient because they can receive all types of blood. Thing to keep in mind, when you are receiving blood, you are receiving just the red blood cells. They will separate the plasma out of. So you're not getting the plasma antibodies, you're just getting the red blood cell. So this red blood cell right here, this person could receive. This individual is type B. So the only marker they have is the B marker. So as you can see, it's got the little green circle, but not the triangle. They have, in terms of plasma antibodies, they have anti-A. That's what these purple structures are showing. What type of blood can they receive? They can receive type B blood and they can receive type O blood. Why can they not receive type AB blood? Is because look right here, this has a marker for this A on it. If this individual type B received this little purple antibody, it's going to combine with that marker and it will start to clot and then lyse. And you don't want that happening. You don't want that clumping to occur in your blood vessels. That will end up killing you. We continue on. An individual who is type A has the A marker. They have anti-B in their plasma as the antibodies. They can receive type A and they can receive type O. And then type O, they do not have any of those markers on the surface of the red blood cell. So they are known as a universal donor. Remember, it's the red blood cell that is being donated. When you receive blood, this is what you're getting. There's no markers on it, so everybody else can receive it, so that's why they're known as a universal donor. However, a person who is type O blood, they have anti-A and anti-B. They have both the green and purple antibodies in there floating around in the plasma, so it could combine with everybody else's. So they can only receive type O. They can give their blood to anybody, but they can only receive type O blood. When you talk about the RH factors, RH group, this is another type of marker that may or may not be present. It's determined genetically once again. We call it RH because it was originally discovered in rhesus monkeys. In a lab setting, sometimes it is also referred to as D. They use the letter D. Now, this is Americans, but overall, globally, 85% of the world population is Rh positive, meaning that they have this marker. So that means 15% is Rh negative. The problem comes when you mix Rh positive blood with Rh negative blood. The first time you're ever exposed to it is not a problem. The person who is Rh negative, that first time it's kind of like ooh, there's something different here. And you're going to start to make antibodies against it. And so any subsequent exposure, those antibodies are going to bind with it. This becomes a problem, especially when you have a woman who is Rh negative and she is pregnant and the developing fetus is Rh positive. If it is her first pregnancy and she has never had a blood transfusion, has never been exposed to Rh positive blood, there will not be a problem. Now they will monitor her very closely, and there are shots that they can give, usually around week, I believe it's 28, and then at the time of delivery, to try to tie up so she does not produce antibodies against it. But they will monitor her closely. There is potential for problems in subsequent pregnancies because now she has made antibodies to it, that Rh factor, and she has another pregnancy and the developing fetus is Rh positive. During pregnancy, the mother's blood and the developing fetus blood does not mix. However, proteins, and antibodies are proteins, can cross the placental barrier. And so those anti-Rh antibodies, those proteins, can pass from the mother into the developing fetus and start to break down the blood with devastating effects. So they will monitor it very, very closely. There are ways that they can give certain medications to try to prevent it. So that's why they will monitor it closely. And I personally have known, I have some very close friends who are Rh negative, who have had very successful multiple pregnancies with Rh positive children. So if you are Rh negative, I'm not saying you cannot have a very successful pregnancy with wonderful outcomes. I'm just saying they will monitor you closely for this. How do you test the blood? What you're doing is you would take drops of blood, and then you add an anti-A serum and an anti-B serum, and you look for clotting. So in this case, what you're trying to see is, what do they have? Do they have those markers on the red blood cells? And so if you see clotting for both, that would be type AB. If you see no clotting for both, then it's type O. If you see clotting here, it'd be A, and if you see clotting here with the anti-B, it's going to be type B blood. So it's relatively easy and very quick and easy to do. But it's very important because if you give the wrong blood type to a patient, it can have devastating effects. It will kill if not caught in time.